Let's see the rearrangement of the heavy chain of the immunoglobulin. Let's put again the structure of the immunoglobulin, and we have already seen it in many videos. This is the constant region of the heavy chain. This is the variable region of the light chain. This is the constant region of the light chain. This is the variable region and constant region of the light chain. This is variable and constant region of the heavy chain, variable and constant region of the heavy chain. We have already seen this structure in many videos, in many previous videos. In red is the heavy chain, in black is the light chain. And as we already know, we have variable region and constant region. This is the variable region of the heavy chain, and all this is the constant region of the heavy chain. So right now we are going to see how the plasmatic cell, this is the plasmatic cell, how the plasmatic cell, when it's synthesizing immunoglobulins, how it's going to make just the heavy chain. In the next video is how the plasmatic cell synthesizes the light chain. But right now we are just going to see the heavy chain. So let's erase. And we need to put the DNA. Right now we are just going to see the chromosome 14. So all this is the double-stranded DNA of a plasmatic cell. We know that plasmatic cells are the cells that make antibodies. And so this is the chromosome 14. So we have, here is the cell, and we know that we have 23 pairs of chromosomes, but I'm not going to put the 23. I'm just going to put the number 14 chromosome. Number 14, and we have the paternal chromosome and the maternal chromosome. I'm just putting the 14, and I'm just going to pick one. Right now, I'm just going to pick one chromosome. So this is the whole chromosome 14, and we're going to divide the DNA, the double-stranded DNA, in segments. So in red, I'm going to put the B segments. These are B segments. So let's call this B1, B2, B3, B4. Then we have many nucleotides. We know that DNA is made of nucleotides. So right here, there are many nucleotides, and we continue with these segments. So this is D1, D2, D3, and now let's continue with J segments. J1, J2, J3, and so on. We just have B and D and J segments, but we have a lot of them. I will just put 4 here, 3 here, and 3 here, but we have literally a lot of them. And here we have something that is called C, and we can have mu, or C delta, C epsilon, C alpha, or C gamma. So we know that this is DNA, and we know that DNA is transcribed into RNA. So from this DNA, we're going to synthesize some RNA. This RNA is going to mature. We will review a little bit about the maturation of the RNA, but then this RNA is going to mature, and the RNA then is going to be translated into proteins. So this RNA is going to enter to ribosomes, and the ribosome is going to synthesize a protein. Which protein is going to synthesize? The heavy chain of the immunoglobulin. So we are going to have something like this. The structures that we have previous, but I'm just putting the heavy chain. And we already see that we have the variable region of the heavy chain, and constant region of the heavy chain. So how we're going to end up with this? So let's see, we need to synthesize RNA from this DNA, but it is not going to be the whole DNA. All this is a gene, and we know that genes are transcribed into RNA, but we are not going to transcribe the whole gene. We are going to take away some segments. We need to pick just one B, just one D, and just one J segment. So all the other segments are going to be removed from the DNA. So let's put it by steps. Step one, we need to pick a D. Even though we begin with B segments, we begin choosing the D segment. So let's pick D2. So we put again the whole RNA, and we just remove these segments that we didn't choose. If we choose the 2 segments, we need to remove the whole these segments that are not the segments that we chose. So let's put again, here is the DNA, here is the DNA. We have again all these segments, so B1, B2, B3, B4, B1, B2, B3, B4. We have just the segments that we choose, that it was the 2. Then we have the whole yes, so yay. J1, J2, J3. Then we have a lot of nucleotides, and we have all these. We need to put all these. Later on, we are going to pick just one of these segments, but right now we need to put all. So C mu, C delta, C epsilon, C alpha, and C gamma. Step 2, we need to pick one B segment. So we pick one D segment and we remove the D segment that we didn't choose. Right now we're going to do the same thing but with B segments. So let's pick, let's say B1. So we're going to remove the B segments that are not the B that we chose. So again we put the whole DNA. We just put the B segment that we chose, so in red. B1. Then we have the segment that the D segment that we chose, that it was D2. Then we need to put the whole J's, so J1, J2, and J3, and we need to put again all these. So I'm just going to put them here. C mu, C delta, C epsilon, C alpha, and C gamma. Right now, we continue having DNA. This is DNA, and we have just removed some pieces of DNA, the D segments and the B segments, but we continue having DNA. This is DNA. Next step is to transcribe this DNA into RNA, even though we haven't choose the J segment. So even though we have here all the J's, right now we're going to transcribe the DNA into RNA. So here is RNA polymerase. Here is RNA polymerase, and it's going to read the DNA and it's going to synthesize RNA. So right now, this is a single-stranded RNA. And we have, again, the B segment, B1 segment, D2 segment. We have the whole G segment, so J, J1, J2, and J3, so on. And we continue having all these segments. So C mu, C delta, C epsilon, C alpha, and C gamma. This is RNA, but remember that RNA needs to do some steps in order to get mature. This is immature RNA. So let's review the 3 steps of maturation of RNA. So the 3 steps of maturation of RNA are, we need to add some cap here, which is 7-methyl-guanosine. So adding 7-methyl-guanosine. Then we need to put here a poly-A tail. We are going to add many adenines here. So poly-A tail. Here we need to add the 7-methyl-guanosine. So here this is 7-methyl-guanosine, the first step. And the third step is the splicing or the alternative splicing. Alternative splicing. So let's review briefly what is alternative splicing. When we have RNA, I'm going to erase here. So when we have RNA, or first, when we have DNA. When we have DNA and we say this is a gene, if we could see, let's see, inside a gene, we will see that we have here in the gene, we have some segments that are called exons and some segments that are called introns. So in red I'm going to put introns, in black are exons. And let's go intron 1, intron 2, intron 3. In the last step of RNA maturation, this is DNA and we're going to transcribe this to, this is DNA and we're going to transcribe this to RNA. So in RNA, we're going to have the same. We're going to have exons, and we're going to have introns. This was DNA, this was DNA, this piece of DNA, this gene. If we could see, let's see more closely, we will see this exon, intron. This is DNA. Then it's going to come RNA polymerase. Here is RNA polymerase, and it's going to read this DNA and synthesize RNA. The same that we just see here, but let's review this briefly. So we have RNA and we have exons and introns. In alternative splicing, what we do is we remove all introns, literally all introns. So in alternative splicing, we remove all the introns, and we can leave all the exons, or we could take away some exons. Not all, but we could leave all exons or take away some exons. And alternative splicing is we could take away some exons, or we could take away another exons. So let's put two variants. Here we could take away all the introns, that is always we are going to take away the introns. So we take away intron 1, 2, and 3. Here, intron 1, 2, and 3. And we could take away, let's see, exon 4. This is exon 4. So what do we have? We have exon 1, 2, and 3, and just that. So this is RNA. This is RNA. But we remove the whole introns and the exon 4. We remove exon 4. So we have exon 1, exon 2, and exon 3. Let's do another variant. In this same RNA, we could take, again, as always, take away all the introns, but we could take away, let's see, exon 2. So what do we have? We have exon 1, exon 3, we remove this, and exon 4. And remember that we have the 7-methylguanosine, so here is 7-methylguanosine, and we have the poly-A, so here is poly-A, 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 poly-A. So this is mature RNA. What do we do to have a mature RNA? We need to add 7-methylguanosine, that is here. We need to put a poly-A, that is here, poly-A, poly-A. And we need to do alternative splicing. Alternative splicing is take away literally all introns, and we take away some exons. Here we took away exon 4, and here we took away exon 2. So this process is going to be the same. Here, just a little bit more complicated. So here we have the RNA. We already did the first two steps of RNA maturation. That was add the 7-methylguanosine and the poly-A tail. Next step is splicing, alternative splicing. Next step is alternative splicing, and we already see that we could take away some exons. Literally, we are going to take away all introns, but we could take away some exons. So here we took away exon 4. Here we are going to take away all the J's, and we are just going to leave one. So these J's correspond to many exons, and we are just going to pick one J, and that is going to be one of the exons that we are going to leave. So this is exon. The B segment is an exon. This segment is another exon. So let's see, exon 1, exon 2. J1 is exon 3. Let's say J2 is exon 4, and so on. We are going to take away all the exons that correspond to J's, except one exon that corresponds to J. So we are going to leave exon 1, which is B1. We are going to leave exon 2, which is D2. And we are going to pick one exon that corresponds to J. So let's say that we pick this. So all these J's are going to be removed, but these are exons. These are exons. So as always, we need to take away all the introns. Let's say this is intron, this is intron. But we are going to take some exons away, which are all the J's except one. Remember that we said we need to pick one B, one D, and one J segment. Right now we are choosing the J segment, but right now we are in RNA. When we choose the B and the D segment, we were in DNA. Right now we are in RNA. So we have, again here, RNA. We have the B segment, B1, the 2 segment, the 2 segment. We chose this, so J1, and these also are exons, and we are going to just pick one of these. So let's say that we pick this, and all these are exons that we are going to remove. The same way that we remove the J's that we don't pick, we just pick one J, and we removed all the others. Here is the same, we're just going to pick one of these, and we're going to remove all the others. Right now we pick this, so C mu. And right now we have an RNA that is mature. It has the 7-methyl-guanosine, it has the poly-A, let's put here the poly-A, and we already did alternative splicing. What we did in alternative splicing, we removed the whole introns and we removed some exons. Which exons did we remove? We removed all these except one, that it was C mu, and we removed all the J exons except one, that it was J1. So right now we have a mature RNA, and this is going to be read by ribosomes. So here is a ribosome, and the RNA is going to be read by the ribosome, and the ribosome is going to synthesize a protein. Which protein? The heavy chain of the immunoglobulin, or of the antibody, which is something like this. The structure that we see at the beginning of the video. And we said we have a variable region, which is this, and a constant region, which is this. This is variable, this is constant. When the ribosome is reading from B1 to J1, is adding amino acid and amino acid and amino acid one with another. But all this, when ribosome is reading from B1 to J1, is synthesizing the protein, or is adding amino acids that corresponds to the variable region of the heavy chain. And when ribosomes read from here to here, is going to add amino acids that corresponds to the constant region of the heavy chain. So right now we have a heavy chain. Then we need to synthesize the light chain that is in the next video. And when we have a heavy chain and a light chain, we are going to join them. But right now we just see the rearrangement of the heavy chain. Why this is called rearrangement of the heavy chain? Because we have we could choose one B, one D, and one J, but just one, and we could choose the one that we want. We could have chosen B1 with, here we had B2 and J1, or B2, D2 and J3. We could have chosen the one that we wanted. It doesn't matter. But obviously if we chose another segment, right now we choose B1, B2 and J1. But let's say that we would have chosen B3 and D2 and J1. We just changed B1 for B2. If we would have chosen B3, obviously this corresponds to sequence of nucleotides, and obviously this sequence of nucleotides are different from this sequence of nucleotides. That means that when we are here in RNA, obviously the nucleotides that we chose, that be B3, the B3 segment, are different from the B1 segment. So if they are different, obviously when the ribosome is reading the RNA, is going to read other nucleotides, and if the ribosome reads different nucleotides, it's going to add different amino acids. So the variable region of the heavy chain is going to change. So we could do many variable regions, or many kinds of variable regions of the constant of the heavy chain. We could do many types or many kinds of variable region of the heavy chain, just picking one B segment, one D segment, and one J segment. And if we change the B or the D or the J segment, it's going to change a lot. So because we could pick one B, one D, and one J, but we can pick the one that we want, or the one that we want, if we speak like teleologically, we could have a variable region of the heavy chain that is going to recognize some antigen when the variable region of the heavy chain binds to the variable region of the light chain. That complex, the complex of the variable region of the heavy chain with the variable region of the light chain, this has a perfect structure that it can just bind to some antigen. Let's say this antigen, when we have the variable region of the heavy chain and the variable region of the light chain joined together, this is a perfect pocket to some antigen, to some specific antigen. and this variable region of the heavy chain is specific to just some kind of antigen. If we would have chosen another rearrangement of segments, B, D, or G, if we would have chosen another, this structure would have changed, and if this changed, we are not going to bind to the same antigen. If we would have chosen, instead of B1 and D2 and J1, if we choose B1, D2 and J1, let's say that we have some structures that is something like this. But if we choose, instead of B1, we choose B3, obviously the structure is going to change. So let's see that right now we have something like this. So right now, or in this structure, here cannot bind the antigen. We need to add another antigen. Let's say that right now this structure is perfect in order to bind, let's say, this antigen. So just changing the B or D or J segment that we're going to pick in order to make the variable region of the heavy chain, we're going to have different iotypes. In other video we already review the term of iotype, but we're going to have a different iotype if we chose different, or if we choose different B, D, or J segments when we join the variable region of the heavy chain with the variable region of the light chain. Depending on which segment we chose, we are going to have some structure. We could have this structure of the heavy chain. If we change the segments, we are going to have another structures. And when we combine the variable region of the heavy chain with the variable region of the light chain, we are going to have some structure, some pocket that could bind to just one kind or one specific antigen. If we change the structure of the heavy chain, it's going to join to another kind of antigen. So this is the rearrangement of the heavy chain. This is how we synthesize a heavy chain. And we see that we need to pick some B, some D, and one J segment. Is in that order. Is first D, then B, and then J. And when we pick the B and the D segment, we are in DNA. When we pick the J and when we pick some of these segments, we are in RNA. And it's in the step of the alternative splicing. See you in the next video.